Okay, hello. Welcome back after lunch for 2 more sessions. We'll start with Martin Jones from the Francis Crick Institute, and he will tell us about making modern computational technologies accessible for volume electron microscopy. Martin.
Okay, thank you. Let's see, is that gonna work? No. The button's not working. Is there enough? Okay, hang on. Okay, perfect. Thanks for the opportunity to present our new proto CCP. So this is one of these 2-year scoping projects, so funded from January this year until end of next year, and we're looking at establishing the need for a collaborative computational project for volume electron microscopy. As part of this, so far, we've been surveying the community. We just recently got the results of that, which we'll be publishing soon. And we had a big community event with about 80 attendees hybrid about a month ago. And what I'm going to show you is the sort of proposals that we have for how to help this community. So first of all, for anyone who's not familiar with electron microscopy, so you've probably all done microscopy at school. Here we're using electrons instead of light, and we can really see smaller objects than light microscopy. This guy is an amphipod, lives in a pond. Scale bar here, this thing's about 2 millimeters. With an electron microscope, we can keep zooming in. This is a diatom, which is a type of algae, and we can keep zooming. And this is a single bacterium, which is now of the order of about 1 micron in size. With a light microscope, this is about the pixel size that you get for this. So this is the reason, in a nutshell, why we use electron microscopy. There's a few different types of electron microscope that we use. Historically, 2 broad types, scanning EM, where we have a sample, we focus a point-like beam of electrons down, bounce them off the surface, scan that point across the surface to build up an image. It gets these sort of characteristically beautiful images. This is a single type of cancer cell. It is about 20 microns across, and it's really looking at the surface of the sample. The other broad type of electron microscope we use is transmission EM, where we now have the sample in the middle and we shine essentially a torch beam of electrons through and we look at the shadow behind it. To do that, we need to embed this cell in something super hard like resin or ice. We cut a very thin slice through it. Electrons don't like to go through much stuff, so this has to be less than a couple of 100 nanometers thick. The electron microscope needs to be under vacuum to keep the air molecules out of the way. This is the sort of typical image you get from one of these. So this is the inside of a cell. This is a dividing nucleus. You've got organelles like mitochondria and endoplasmic reticulum. This is looking at the inside of the sample, but both of these are essentially 2D or projected 2D. And it can be very risky to make conclusions based on a 2D view. You really need to understand the 3D context of what you're looking at. So for this, we have this field of volume electron microscopy, or VEM. This is a particular type. There are very many different ways of doing this. This is serial block face scanning EM. We build up the image by scanning the point like a regular scanning EM, but now we have a diamond knife in the microscope that cleaves away the top few tens of nanometers. Different types, so that produces this kind of image. So this is scrolling slice by slice, 50 nanometers at a time through each of these as an individual cell. This is from a different type of volume electron microscope called a focused ion beam scanning electron microscope. And this is going through 5 nanometers at a time, or even down to 3 nanometers at a time. So the data sizes for these, for an acquisition of a day or a few days, can be up to terabyte scale quite quickly. And the rapid advance in these technologies is one of the reasons why volume electron microscopy is one of nature's 7 technologies to watch in 2023, alongside the James Webb Space Telescope. So we have these automated instruments that acquire tons and tons of really cool images. Very often they're kind of tile scans, so we have to stitch them together, and we have to align these slices in Z, so we make a 3D volume. And configuring this is very hard for non-experts. There's lots of different tools that claim to do it. Once we've got the tiles stitched together, then we take our different Z slices and align them into some sort of spatially coherent stack. And then once we've got to here, now we can start to analyze the data. The most common sort of analysis we do is something called segmentation. So that's the process of separating the object of interest from stuff you're not interested in. So here the flashing green line is the segmentation of the nuclear envelope, the membrane around the nucleus. And if we do that in 3D, then we can arrive at something like this. And now we can start to measure things. So did that drug treatment change the volume of the nucleus or change the positions of the other organelles within the cell? Currently, this is done almost entirely manually. So this involves people sitting for days, weeks, months, or sometimes years, going through data sets that you maybe acquired in a few days. So lots of information is left on the table. We can't mine all of this using these manual techniques. So AI to the rescue. There's a lot of nice AI methods out there which we're making use of. So convolutional neural networks we've heard about already earlier. These are very well suited for these vision processing type tasks. Some of the new vision transformer models and various hybrid classes between the two work very well as well. If we can get to the stage where we're analyzing very many of these objects, then we can start to do statistics, and we can be very, we can interpret the information that we acquire in these images much more thoroughly. Training and using AI is still very challenging for the end users in the lab. So most biologists don't do maths, computing, or physics beyond kind of GCSE or A-level. So these are very challenging to even present them with like a Jupyter notebook to do this kind of stuff. So anything we can do to make this, using these tools, easier will have a huge benefit for the field of biology and biomedical imaging. So using this AI model, here's a bunch of nuclei that we extracted from that volume. Now we can start to do our statistics over these different samples at scale. So that's where we really want to get to. Okay, so the current situation, there's quite a lot of these data sets out there. They've been relatively lightly mined for information, but they are incredibly rich in information. So one of our goals with the CCP is to employing these FAIR data principles, so findable, accessible, interoperable, reusable, make these data sets available to people who might be able to mine useful information out of them, maybe information that we didn't think about, maybe train future AI models on these things. There's a lot of tools out there. There's some nice solutions. We don't necessarily need to invent new neural network architectures to do this, but at the moment there's analysis paralysis. People don't know what to use, how to use it for their tasks. People see things published. I'd love to use that, but I couldn't install it. I can't work out how to use it. I'm not really sure if this is the right tool. Will it work next year? These are the sorts of comments we get when people try to use these tools. So this is most people's response. Okay, we'll get the PhD student to write something that does it. This is sometimes great, sometimes very often leads to these kind of unsustained software packages that kind of fall a bit by the wayside, and we get this kind of disjointed and fragmented ecosystem that makes interoperability very tough for these analysis pipelines. So what we propose with CCP-volumeEM, notice we went straight for CCP, we're very bold, we didn't go for CCC like some of our colleagues. We're hoping that we get through to a true CCP after our probationary period. And we kind of identified these three work packages to work on as part of the CCP. So a big part is community building and consolidation. There's a lot of people writing software, getting data, microscopists, analysts out there who don't necessarily talk to each other yet. There's obviously a research software element about producing good, robust software. And then there's a huge training element. I think we see this as one of our major endeavors. So there's a huge number of stakeholders in the sort of volumeEM biomedical imaging. So it kind of came from something called the volumeEM initiative, which is sort of a community that was sort of funded by Chan Zuckerberg Initiative for a few years and is now sort of largely voluntary, I think. There are a lot of microscopy organisations, Royal Microscopical Society, Bioimaging UK, a few image analysis organisations like New Bias, ImageSC, Glow Bias. Of course, CoSeC, we're now very grateful to be involved with CoSeC on this. So there's a lot of people coming at this from lots of different angles. And like I say, we want the community to recognise this fact that there's this huge amount of rich data out there in these archives. Secondary use is possible. People who don't really care about the biology can use this data to train their AI models. To do that, we've heard it a couple of times already that some sort of sensible metadata standards are going to be really important for this. So in biology, just in the last couple of years, there's an initiative called REMBI, which is recommended metadata for biological images, which has tried to create something of an ontology for how the different stakeholders, so the biologists who care about the drug treatment or the cell type, the imaging scientists who set up the resolution and the microscopes, and the computational researchers who develop the algorithms, they all need different kinds of information to be able to join this up and really get the maximum benefit out of these data sets. So this REMBI initiative is really trying to cover all of that so that we future-proof these data sets for these kind of long-term uses. There's a lot of technical barriers. A lot of our end users struggle to use anything that's not sort of commercial polished software. So even things like installing software is a challenge. We're identifying that containerization is potentially a good way of getting certain types of software into the right hands without people having to worry about Conda environments and such. Lots of caveats to that, of course. We're working on this. This is kind of the gold standard for reproducibility. So increasingly now people submit containers with their publications because that is in principle guaranteed to produce the same results that you showed in your paper. And the idea that you can run it on multiple different platforms is appealing because everyone in biology has very varying computational infrastructure, I think is fair to say. And this is work that colleagues at the Rosalind Franklin Institute have been working on. So Gabriel and Piper have worked on generating a lot of these domain-specific containers that we can run across multiple platforms. But like I say, this is very far outside of biologists' comfort zones. We mentioned this at our community meeting and most biologists looked very scared. So we're trying to work out the best way to make the best use of this without unnecessarily complicating things. A big technology that we're really pressing is this thing called next generation file formats. So if I have a regular image stack, so here's a kind of toy example of 20 slices of 10 by 10 images. And if I want to analyze some subset of this volume, so down here, I'm interested just in this little red cube. I have to load a whole bunch of data that I'm not really interested in into memory to be able to process it. And this, for anything kind of realistic, becomes quite prohibitive. Individual slices can be really huge in volumeEM. With these next-gen file formats, for example, OMI-ZAR, which is kind of a microscopy overlay of the ZAR format, it uses this kind of more chunked format. So now, instead of whole big monolithic slices, now we can chunk down to some particular size. So here, 2 by 2 by 3. And now I can random access chunks through this data set much more efficiently. So now this small region I want to capture here only requires me loading much less data into memory, and it's a much bigger advantage in realistic data sets than in this toy example. And it allows us to load data sets that are much larger than available RAM. Also, these Czar imaging formats tend to use this kind of Google Maps style multi-resolution map organization. So we have multiple resolutions stored within the Czar hierarchy. And what that allows you to do is, depending on where you're zoomed into on your screen, this is a 2.5 terabyte data set that somebody's scrolling through tethered on their phone. The system only gets the pixels that it needs to lay onto your screen and nothing else. So it can be very efficient to handle very, very large data sets. And this is great for visualization, but it's not really been leveraged for analysis so much yet. So that's something we're trying to do now with our software engineering aspects, is working with the community developing these tools to create some kind of exemplar workflows. So really over-engineer, build out workflows, very elaborate documentation, very elaborate commenting in the code to solve a particular problem. We've chosen stitching and stacking these 3D imaging data sets together. So we've taken a publicly available data set. So it's something around 1 terabyte, I think, in total. It's a huge, big tiled, 10 by 10 tiled. Each tile is 6,000 by 6,000 pixels, 70 slices, many hundreds of gigavoxels. And our colleague Joost has been working on using the OME NGFF or OME Czar format, along with latest parallel processing libraries like Dask, to stitch these things together in a way that is essentially impossible at the moment. People do this manually and it takes a really long time. It's not perfect, but now we have a consistent method for building up these analysis workflows. And we really want this, as well as to be a tool that people can use, we want to onboard developers to be able to learn how to implement these methods in their own software. Another thing we're doing is supporting developers. So there's, like I say, there's a lot of really nice software out there. One really nice example is a plugin in this Nepari platform called Empanada, which uses various deep learning pre-trained models to segment different organelles within cells. And it's great. It was published pre-printed a couple of years ago, and it kind of fell into this common problem of not enough developers, not enough time to update it. So it kind of got a bit frozen in old versions of Python, old versions of different libraries, and it stopped being compatible with lots of other things. So Mariam, who's somewhere in the audience, I think, has been working with the Empanada team to bring this up into the latest libraries and tools. And I think a really amazing opportunity of CoSeC and CCPs is that this knowledge then becomes centralized. So the next time somebody wants to do that, they don't have to reinvent the wheel. There's some kind of centralized knowledge how to solve the common problems. Like I say, training is a big part of what we want to do, or what we are doing. Technical setup for training when you're dealing with terabyte data sets is challenging. So we've been working with this data analytics as a service platform, I think now Ada is called, to sort of decouple this installation phase from people learning to use the software, using these containers. So it runs on these virtual machines at STFC, but we can also run them on our local HPC for local trainings. And we have successfully run a couple of courses where we've used these containers on some of these modern software packages to upskill people. The kind of courses we want to teach. So end user courses, so how to use the different softwares, Fiji, iMod, kind of domain specific things. We also want to train developers how to use best practice in developing their tools. So how to do proper testing, how to do proper version control, things like this. When new to the community tools emerge, we want to present those via these training methods, and work with the people who are developing other tools so that we can help them provide training for the community via these platforms. Hackathons, we think, is a useful way of doing some of these more developer-facing trainings. And some of this is not specific to volumeEM, right? So some of these principles are general to all CCPs. So I'm hoping that there's opportunities to work with other CCPs on that. So in summary, sorry, very quick, too many slides. So we've been looking at what the landscape is like at the moment. There were no huge surprises, but we know there are a lot of problems. We really want to get this idea of best practice across and reproducibility. We want to future-proof to use of AI in the future, support tools that people are already using. We don't want to tear people away from what works. We've got a wide range of people we need to train, so we need to cover that well. And like I say, much of this is applicable beyond volumeEM. A lot of people involved. So the top half are people who were on the grants. Martin is our SCFC CoSeC contact, and then everyone below there is people who onboarded to the activities since we started. And I'll finish there. Thanks.
Quick question. Okay, Edo, but quick.
Yeah, it's Let Smith. Sorry. I was wondering about the ePSRC, where you have multiple versions of your data. So it bloats the data, right? There is a cost.
So if you solve
environmental,
if you do the sums, then if you only downscale in XY, then your data gets 1.33 times the original size. If you downscale in X, Y, and Z, it's 1.25, but it compresses very well compared to TIFF. So actually our ePSRC data sets wind up being slightly smaller than the original TIFF stacks, uncompressed TIFF stacks. So we can compress ZAR quite effectively compared to some of the...
But you don't have...
No. So it's very hard to convince biologists to do anything that looks to them like losing data.
Or you can charge them for storage. So when they will...
Yeah, I've tried that argument. It still hasn't worked.
Okay, so let's think... okay, a quick question there. Do you have a question? Yeah.
Just a quick question on what you do with the large image archives. You mentioned bioimaging. Is that the primary target, or do you have some intermediate place where you publish data publicly? But you mentioned also that was very large, which of course limits your options and how to make things available.
I'm quite lucky. So I'm at the Crick, which is a new shiny institute that has quite a lot of resources. So we do have a local HPC. So we do have things on there, and then at the end it goes to Bioimage Archive. So I think probably this sort of intermediate stage is also something to be worked on, because yeah, the data sets get very big very quickly.
Okay, so let us thank Martin again.